Hello, everyone, and welcome to our fifth lecture in our course on general psychology. In this lecture, we're going to begin another module. So this is our third module, and here we're going to talk about the brain. And in particular, we're going to talk about neurons, which, as the title points out, are the building blocks of the brain. So neurons are individual nerve cells. By way of overview, we're going to be learning about neurons. And as I just said, these are the building blocks of the brain. These are the cells that comprise a vast amount of your brain, not all of your brain. As your book mentions, there are also cells called glial cells. We're not going to talk about them very much. They're not as well understood, but they take up a significant volume. Nevertheless, neurons exist in your brain in the tens of billions, so there are lots of them. We're going to start by talking about their physical structure, what they look like, how they're built, what their parts are. And that physical structure is going to inform our understanding of neural function. So these are very special cells, not just for our course, but in the context of your body. And so neural function is based on the structure of neurons. Neurons wouldn't work the way that they do if they weren't built the way that they are. And we're also going to talk about some of the chemical messengers that are involved in neural communication, how neurons talk to one another. And that will also lead us eventually into our unit on psychoactive drugs, which will be in a few weeks. Because psychoactive drugs use chemical messaging sort of the same way that neurons do in their native environment. Psychoactive drugs kind of hijack the nervous system in a sense. By fooling neurons into thinking that they are the brain's native chemical messengers. So in this session in particular, what we're going to do is we're going to learn the different components of a neuron, what the structures are, what they're called. And then once we have that structure in mind, we'll begin to understand how neurons create these electrical signals. It's going to involve a little bit of chemistry, but if it's been a while since you had chemistry or biology, don't worry. We're going to go over the important basics, and there's really just a couple of moving parts for understanding neural function, at least signaling within a neuron. And then we're going to move on to the next step, which is how neurons communicate with one another. So they do that using those chemical messengers. Okay, so of course at first we had no idea that neurons existed. And it wasn't until a Spanish scientist named Santiago Ramon y Cajal discovered that the old view of the brain was inaccurate. It used to be thought that the brain is sort of one big web of tissue. It's one interconnected mass. And while there are connections between parts of the brain, what Ramon y Cajal discovered when he looked at images like this of individual brain cells by staining them, he discovered that the brain is composed of discrete cells. He did some drawings like this one, and these were the first drawings of brain cells, and he proved that the brain isn't just one big continuous lump of tissue, as was previously thought, that messages don't pass through the brain because it's one big cell, so to speak, that rather the brain is composed of discrete cells, separate cells. And that was kind of revolutionary, but it shaped our thinking ever since. And Ramon y Cajal started to develop what is now known as the neuron doctrine. And there are various rules or guidelines in this doctrine. As time goes on, different guidelines get subbed in or out. But the most basic components are, first, that the brain is composed of individual cells, not this big interconnected mass. There are discrete cells, they have their own cellular processes, they have signals within the cells, and so they're individuals. And a follow-up to that is the brain still has communication within it. Neurons talk to one another, and they transmit information across what are called synapses. And synapse may be a word you've heard before, you already may be familiar with it, but synapses are the connection points between neurons. And also there are different types of cells. So not only is the brain not one giant interconnected mass, there are different kinds of cells that talk to one another. And those cells vary in terms of their structure, and because they vary in structure, they're going to vary in function. And different parts of the brain have different kinds of cells, as we'll see. So how are neurons built? So I'm going to show you here a basic diagram of a neuron. As I just said, neurons come in various shapes and sizes, so we don't want to make the mistake of thinking that all neurons look like this one. But this is a fair depiction of a typical neuron. And the thing about neurons is they are information processing units. And each part of a neuron deals with that information processing. Each part of a neuron has a different job. So you have the dendrites, which are up at the top of the diagram on the right, and they bring information in. So they receive information from other neurons. And that's their main job. You can see there are lots of them. They have these spine little projections because neurons can connect, can form synapses with a large number of other neurons. So it's not a neuron just receives information from one neighbor. It can receive information from many neighbors. And so that's what dendrites do, is they bring in information, they receive information. Then you have the cell body, sometimes called the soma. And the cell body sort of coordinates tasks. It has the nucleus of the cell, so it contains the DNA. But the cell body is sort of the central junction. It's where all the points connect. It's also where all that information will come in and get collected. And then it's also what maintains the cell. A lot of the cellular machinery that's found in most cells, a lot of that machinery is just involved in keeping the cell alive. And so a neuron is specialized, but it is still a cell. So it's alive. It has parts that need to be maintained. It needs to make proteins. It needs to take in energy and use that energy. And so it has a lot of those moving parts that all cells have, and we're not going to go into details there, because what we really care about with neurons is their ability to process information. Then you have the axon, and so the cell body collects information, and if it's going to generate a signal, then it sends that signal down the axon, and the axon is the lowermost portion of the neuron in the diagram on the right. So the axon transmits information. Dendrites bring in information. Axons transmit information to other neurons, sometimes to muscles or other organs, mostly to neurons, especially in the context of the brain. As you can see here, also, the axon is covered in what's called a myelin sheath. And that is, as the name suggests, it's a covering made of a substance called myelin. And myelin is a fatty, sort of waxy substance. And what it does, you can see here on the right, it sort of makes the axon look like a series of sausage links. But the purpose is to make information transmission faster and more efficient. Not all neurons have myelin sheaths. Some lack them. And those that lack them are mostly in the spinal cord and the brain, and those are the sorts of neurons that talk to one another locally. If an axon has a myelin sheath, odds are it's transmitting information a long distance. The myelin sheath is also the part of the neuron that gets degraded in diseases like multiple sclerosis, MS. So if you lose that myelin sheath on a neuron that's supposed to have one, that can really damage that neuron's ability to communicate. And then finally, the axon ends in these little branches that you can see. And those are the axon terminals. Those are the parts of the neuron that actually connect to the dendrites of the next neuron. And so the axon, there's just one of them, but it can have these little branching points, these axon terminals, that can connect to a large number of other neurons. So a single neuron can receive information from a lot of its neighbors and can send information to a lot of its neighbors. So we shouldn't think that it just talks to one neuron or sends information to one neuron. The axon is capable of talking to a lot of different neurons. The axon terminals are sometimes called terminal buttons or synaptic boutons. I'll always refer to them as axon terminals. Okay, so now we know the various parts of a neuron. And as I've said, neurons connect to one another so that they can communicate. And those connections are called synapses. And a synapse has two neurons in it. It has the presynaptic neuron, and it has the postsynaptic neuron. And that pre and post prefix is important because it indicates the flow of information. So anytime you have a synapse, you're going to have axon terminals connecting to dendrites. Information is going to flow from those axon terminals to the dendrites. If we remember, dendrites take in information, axons send out information. So the presynaptic neuron is the one with the axon terminal, and the postsynaptic neuron is the one with the dendrite. Now, obviously, the one with the dendrite will be the presynaptic neuron for other synapses that it has with its axon. But for a given synapse, there are just two. There's the presynaptic and the postsynaptic. So presynaptic is the one sending information. Postsynaptic is the one receiving information. So here in this diagram, I've circled a few of these synapses, these connection points. And we can see how the axon terminals of the presynaptic neuron connect to the dendrites of the postsynaptic neuron. And it can have multiple connections, even between the two of the same neuron. And if we zoom in, we see this bigger inset here. And what we're going to see is that synapses, the vast majority of synapses in the brain are chemical in nature. What do I mean by that? I mean that the axon terminal, even though the neuron itself uses electrical signaling, that a synapse uses chemical signaling. And so the presynaptic neuron contains chemicals that, when an electrical signal occurs, the axon terminal will release those chemicals, and those chemicals will connect to the postsynaptic neuron via what are called receptors. We'll talk more about that in a minute. But the point is there's a little gap between the presynaptic and postsynaptic neuron. That's part of what Ramon E. Cajal discovered. And so that gap is where chemicals are released by the presynaptic neuron, and it's where they're picked up by the postsynaptic neuron, and that will initiate a signal in that postsynaptic neuron. So if we zoomed in like we have here, we see these chemicals that are housed in the presynaptic neuron released into the synaptic cleft, the gap between the two neurons, and picked up by the postsynaptic neuron. And this cartoon may look complex, but it's relatively simple compared to reality, where a single neuron can form thousands of synapses. So it can receive information from thousands of neighbors, or from a single neighbor many, many times over. And it can also send information to thousands of neighbors, or the same set of neighbors many times over. So you get a lot of complex patterns of connection between various neurons in the brain. As Ramon E. Cajal's doctrine indicated, and as your book says, neurons are also specialized. So there are different kinds of neurons. You can classify them by structure. So here we have a diagram from your book, and we can see how varied neurons can be. They've color-coded dendrites and axons here. You have, for example, Purkinje cells in the leftmost part of the diagram. You have pyramidal neurons in the middle, and you have bipolar neurons on the right. And the point here is not to memorize the fine structures of these neurons. We will eventually be touching back to different kinds of neurons. But the idea is that these neurons are found in specific locations. Now, your book makes it sound, I think, like neurons, there's a one-to-one mapping between a specialized neuron and a location, as though you'd only find a particular kind of neuron in one particular part of the brain. Really, especially neurons like pyramidal neurons, can be found in multiple places. So it's not the location really that determines what a neuron is called, it's its structure. Now, as it happens, a lot of them, like Purkinje cells, are only found in one particular spot. But we don't want to make the mistake of thinking that there's a one-to-one mapping between certain kinds of neurons and locations. It's really the structure of the neuron that determines what they're called. You can also classify neurons by what they do, by their function. So here we have an example of a simple reflex arc. And so you can have, for example, a sensory neuron. And a sensory neuron does just what it says. It connects sense organs, like pain sensing organs, or pressure or vibration sensing organs in the skin, and it takes those signals, those events in the environment, and sends them to the spinal cord. So that's what a sensory neuron does. It transfers information from the body to the spinal cord. You have motor neurons, which kind of do the opposite. They take information from the spinal cord, and these will be commands to move muscles, and transfer those signals to the relevant muscle group. So that way the muscle will contract. That's how we move around. Motor neurons sending signals to our muscles to cause them to contract. And sometimes you have middlemen, especially in the brain, where the vast majority of neurons in the brain are not sensory or motor neurons. They're what are called interneurons. Inter means between. And what interneurons do is they relay signals from one neuron to another. In the simple reflex arc, it's from the sensory neuron to the motor neuron. So you get a pain signal carried by the sensory neuron, transferred to the motor neuron by the interneuron. And the motor neuron causes the hand to withdraw from the painful stimulus. But the brain uses lots of interneurons. That's how you process information. Neurons talking to other neurons. Now, before we get to communication across multiple neurons like that, we first need to talk about signaling inside a neuron. So neurons use electrical signaling. The signals that you would record, you can measure from a neuron, are based on electricity. And in this case, what electricity is the movement of charge. That's what electricity is defined as. Now, in electrical devices or in the wall, you have the movement of electrons. And electrons are charged particles. They're subatomic particles that make up an atom. In this case, the electrical charge is the movement of whole atoms. So when we're talking about the neuron, we're talking about whole atoms moving around, and these atoms carry charge. And a charged atom is called an ion. So just a quick refresher on chemistry in case it's been a while. Ions are atoms in which the number of protons and the number of electrons aren't equal. So protons are found in the nucleus, they're positively charged. Electrons sort of swim around the nucleus. They orbit it, but not in a systematic fashion like diagrams also often make it seem. But electrons have a negative charge. And so if that charge is balanced, if the same number of protons is balanced by electrons, then you have a neutral charge. It's not an ion, it's just an atom. However, if the ion gains an electron, it now has more negative charge than positive. If it loses an electron, it has more positive charge than negative. And then it's an ion. It carries an electrical charge. And that's the sort of atom that we care about, and it's the sort of atom that neurons use for their electrical signals. Now, for our purposes right now, there are two important ions, just two. They're different elements. One is sodium, which we've all heard of, and sodium is found to be plentiful outside the neuron. There's lots of sodium outside the neuron, very little inside. And sodium carries a positive charge. There's also potassium, which you've probably heard of. And this also carries a positive charge, but in terms of location, it's the opposite. There's a lot of potassium inside the neuron, very little outside. And when it comes to ions, and what governs whether they're in one spot or another, there are really two forces that drive their movement. One is diffusion. And what diffusion is the tendency for ions to move from where they're concentrated to where they're not. They go from where they're common to where they're less common. And so if you have a whole bunch of ions in one spot, a lot of those ions will tend to move to a spot where they're less common until everything's balanced out, until the ions are at equilibrium. The other force is electrostatic. And all that means is that like forces, or like charges, that is, if they're both positive or both negative, they tend to repel one another. If they're opposite charges, they tend to attract one another, just like the opposite ends of a magnet, for example. So here, we don't have any negative charges to worry about yet. It's just two positive ones. And so sodium ions tend to repel other sodium ions, but sodium and potassium ions will also tend to repel each other because they're all positively charged. So when it comes to electrostatic forces, sodium doesn't care whether a nearby ion is sodium and potassium. All it cares about is that it has the same charge. And so that will repel the sodium ion. Same thing for potassium. Potassium will repel other potassium ions. It will also repel other sodium ions because they're all positively charged. And those two forces are really going to dictate how electrical signaling works in the neuron. And so that brings us to the electrical signal within the neuron itself. And that signal is called an action potential. Now, let's break that phrase down a little bit. Potential here refers to voltage. And voltage is a difference in charge. We were just talking about sodium and potassium. And there are different amounts of sodium and potassium on the inside and outside of the neuron. We just said that there's lots of sodium outside, very little inside, and lots of potassium inside and very little outside. There's some other ions at play here and some other molecules that determine the charge inside and outside the neuron. We're not going to worry about them very much. But what ends up happening is when the neuron's not really doing anything, when it's resting, it is at its resting potential. And that is the difference in charge when the neuron isn't signaling. And that difference in charge is measured in millivolts, thousandths of a volt. So as it happens, neurons, for the most part, have a resting potential of minus 70 millivolts. What that means is, just by convention, the charge on the inside is compared to the charge on the outside. So inside minus outside is minus 70 millivolts. What that translates into is the inside of the neuron is more negatively charged than the outside. We've already talked about sodium and potassium, but based on their concentrations and those other ions we're not going to worry about, a neuron is usually sitting there at minus 70 millivolts. And you can measure the difference in voltage between the inside and the outside over time. So if you're measuring that voltage with what we call an electrode, what you're going to read out, what you're going to measure, is the neuron sitting there at minus 70, same negative charge on the inside relative to the outside. And that's what I've circled here in blue. So normally, this is the state of the neuron. It's at minus 70 millivolts, and embedded in the membrane of the neuron are what are called ion channels. And these are holes in the membrane, molecules that form little channels through which certain ions can pass, or not if they're closed. And normally the channels are closed. There are a few that are open that are letting potassium through. We're not going to worry about that. But for the most part, the channels are closed. You have lots of sodium outside, lots of potassium inside. There is a little molecule called the sodium-potassium pump that keeps things that way. It keeps sodium out and keeps potassium in. So that's the normal state of affairs. Minus 70 millivolts, lots of sodium outside, lots of potassium inside. Then we get to the action potential. And this is an electrical signal conducted along the axon. And so what we're talking about here, this electrical signal, we're really talking about what's going on in the axon once a signal starts being generated. And what the basis of the signal is, it's going to be the movement of sodium and potassium across that cell membrane after it reaches what we call threshold. The axon takes a little bit of stimulation before it will generate a signal. Now, normally this is because the soma, the cell body, has collected information from the dendrites in the form of electrical charge, added it together, and if there's enough positive charge inside the cell body, that's when the axon generates an action potential. So it's sort of an all or nothing. Action potentials are all or none. They are not a graded response. They either happen or they don't. And they happen or not depending on what's going on in the cell body. So it's sort of a democratic system. All the dendrites are sending information, the cell body adds it all together, and either votes yes for an action potential or no for not a signal. Uh, and so if a threshold is reached, what the threshold is it's a minimum change in voltage. So normally the neuron's at minus 70 millivolts. If it goes up by about 20 millivolts, depending on what's going on in that cell body, then the neuron will hit threshold, and the axon will generate an action potential, which means sodium is going to move in, potassium is going to move out, and the timing here matters. So as I've already said, there's lots of sodium outside. Diffusion, the tendency for ions to move to where they're less concentrated, pushes sodium inside the neuron. Now potassium, if it's allowed to go through the membrane, will move out of the neuron because there's so much inside, diffusion pushes it out. Also, potassium, as we're going to see, and sodium both move away from positive charge. So what happens with an action potential? Well, first you reach threshold, which is a little bump you see on the curve in the background of this diagram. So we see that sort of smooth curve that is the action potential. It is the change in voltage over time in the axon. So what happens first is that when you reach threshold, that threshold is the voltage at which those sodium channels are going to open up. They're sensitive to the voltage across the membrane. When the voltage goes up by 20 millivolts, those sodium channels, sodium moves through its own kind of channel, and so those sodium channels open up when threshold is reached. And now there's all the sodium outside, very little inside, so it comes rushing in. And what that does is change the voltage across the membrane. Remember, sodium has a positive charge. So sodium comes rushing in, it changes the amount of charge inside the neuron, and so we see in this curve that the curve representing voltage goes up. Now, parts of the curve are hidden by the pictures on this diagram, but we see this sort of spike, this hill that represents an increase in voltage. And that's because all the sodium is coming in, and sodium is positively charged. So now that inside of the neuron isn't so negatively charged anymore. It's not at minus 70. It's not even at minus 50 where threshold is. It will actually get into positive territory. You'll have more positive charge inside the neuron than you do outside. So that's sort of step one after reaching threshold. Sodium comes in. After that, potassium channels open up. So potassium channels are ion channels that are sensitive to voltage and allow potassium to pass through. Now, because there's so much potassium inside the neuron, potassium wants to get out. And also, because all the sodium is now inside, there's now positive charge on the inside, and that also drives potassium out. That involves both our diffusion force, potassium going from where it's more common to where it's less common, and our electrostatic force, positive charge pushing potassium away. So both these forces are pushing potassium out, and that's what happens. And this is that downward going part of this curve. So when sodium comes in, the curve goes up because more positive charge is coming in. When potassium starts leaving, the curve goes down because positive charge is leaving the inside of the neuron. The inside of the neuron is getting more negative. And so much potassium leaves that you actually not only get back to resting potential, you overshoot it a little bit. And so that is the up and down motion of voltage in an action potential. After that, that pump I was talking about and the ion channels being open, let them charge back and forth across the membrane until it reaches equilibrium again. And by that point, the channels have closed. And you're now ready for a new action potential. And that's the step we see here, that the sodium-potassium pump reestablishes those, what we call gradients, those differences in concentration, so that there's lots of sodium outside and lots of potassium inside. Now, shortly after an action potential and during an action potential, you can't have another one. There's what's called the refractory period. And the refractory period is that small amount of time in which another action potential is not possible. There isn't enough sodium outside, there isn't enough potassium inside, and the channels are closing anyway, so there's no way for those ions to move. And so that refractory period is when you can't have another action potential. So action potentials are all or none. They fire at the same magnitude, that curve goes up to the same point, regardless of the intensity of the stimulus the neuron got. It doesn't matter whether threshold was just barely reached or whether threshold was reached and then some. The size of the action potential in terms of voltage is the same. The duration is also the same. It takes about a millisecond, a thousandth of a second. So that is the refractory period of the action potential. Now, when it comes to axons, as I mentioned, some axons are covered in myelin, that waxy substance that insulates an axon. And that myelin actually speeds up the action potential, makes it move faster down the axon. And the way it does this is by insulating parts of the axon, not all of it. The gaps between myelin chunks are called nodes of Ranvier. And this leads to what's called saltatory conduction. Saltatory here has nothing to do with salt. It's from the Latin word to jump. And so what happens is the action potential will sort of jump from one uncovered portion of the axon to the next. And because it jumps, it's going much faster than if there were no myelin at all. Certainly lots of neurons in your brain are unmyelinated, but not the ones where signals need to travel long distances, not the ones with really long axons. So for local signaling and information processing, you don't have myelin. But if it needs to travel a fair distance, if the action potential has a far distance to go, then you do have myelin, and you do have saltatory conduction, which is an increased efficiency of transmission. Okay, so that's signaling within a neuron. What about signaling between neurons? And here, as I've mentioned, axon terminals release chemicals. They hold on to chemicals in what are called vesicles, and these chemicals are called neurotransmitters. So neuro, from neuron, and transmitter because it's transmitting information. This is how neurons communicate with one another. So neurotransmitters are chemicals that transmit information. Now this is sort of a more close-up view of that diagram we had earlier at the synapse. And so we can see the vesicles, these little circles or sacs, within the presynaptic neuron that hold on to neurotransmitter until an action potential comes along and causes it to be released. So when neurotransmitters are released, they're released into that synaptic cleft. Your book calls it a synaptic gap. It's the same thing. And neurotransmitter is released into this gap and then diffuses, same process, goes from where it's more common to where it's less common, diffuses across the synaptic cleft and binds to what are called receptors on the postsynaptic dendrites. And so this is how the postsynaptic neuron receives information. That's how it knows that the presynaptic neuron has fired an action potential. So receptors are molecules that are embedded in that postsynaptic membrane, and they receive neurotransmitters, which I've abbreviated here NT. They receive neurotransmitters, and they have the capacity, if enough of them are released, to cause an action potential or to prevent one. And that's one of the interesting things about chemical signaling between neurons, is that based on the combination of neurotransmitter and receptor, when the presynaptic neuron fires an action potential, neurotransmitter is released, and that event can cause an action potential in the postsynaptic neuron to be more likely, we call excitatory, it excites the neuron, or less likely, inhibitory, it inhibits the neuron, it keeps it from firing. Either one's possible, and we'll talk more about that here in a minute. But that's one of the most important things about signaling between neurons, is that A, it's chemical, so you're releasing neurotransmitters, but B, depending on the neurotransmitter and the receptor, it's not a guarantee that the postsynaptic neuron will fire as well. So the postsynaptic neuron collects all the information together, adds sort of the no votes and the yes votes, the inhibitory and excitatory signals it's getting, and then itself either fires an action potential or not. So it has to add all that information together in the cell body. But this chemical transmission isn't always telling the next neuron to fire. Sometimes it's telling it not to fire an action potential. Okay, so what are these neurotransmitters? We know that they're chemicals that the presynaptic neuron releases and that the postsynaptic neuron receives. Uh, and chemically, we're not going to get into it too much. That's not important. The important thing is that the combination of the neurotransmitter and the receptor determines its effect. And a given synapse will really be involved in just one kind of neurotransmitter. Which means the presynaptic neuron only makes one kind of neurotransmitter. You don't have a bunch of these different chemicals going out at once. But what are neurotransmitters? Well, they're chemicals, and based on their chemical structure, they have different names, and they have different functions in the nervous system. So, for example, the neurotransmitter glutamate is the most common neurotransmitter in the entire brain, and it is the main excitatory neurotransmitter. What does that mean? Well, we've already gone over excitation and inhibition. Excitation is when the postsynaptic neuron becomes more likely to fire when it receives one of these neurotransmitters. So glutamate is the main excitatory neurotransmitter, which means if the presynaptic neuron releases glutamate, the postsynaptic neuron is more likely to fire. It's not guaranteed. It takes some coordinated firing across synapses, but it increases the likelihood of an action potential. And 85% of your synapses across your entire brain are what we call glutamatergic. They use glutamate. Its opposite is called GABA, and GABA is an acronym. We don't need to go into what GABA stands for, but it's the opposite. It's the main inhibitory neurotransmitter. So when the presynaptic neuron releases GABA, the neurotransmitter, it causes the postsynaptic neuron to be less likely to fire an action potential. It inhibits the next neuron from starting a signal. And on the next slide, we'll mention drugs a little bit. And some drugs work through the GABA system, for example. And so they'll help to depress activity in the whole brain, and these tend to be depressants. They tend to calm you down or put you to sleep. So that's what GABA does. It keeps neurons from firing. Acetylcholine was actually the first neurotransmitter that was discovered. And it is involved in muscle function, so making muscles contract, and also memory. So depending on where you're looking, acetylcholine, if it's released onto muscles, will cause those muscles to contract. But inside the brain, acetylcholine synapses tend to be involved in memory. And so drugs that counteract memory loss, things like Alzheimer's drugs, tend to work through the acetylcholine system. Dopamine is another famous one. You've probably heard of dopamine. Dopamine in the brain is involved in motor control. So not out on the muscle end of things, but inside the brain, when the brain is trying to initiate movement, dopamine is involved in that process. It's also involved in reward processing. Now, dopamine is historically, and a little simplistically, been described as the reward chemical or the pleasure chemical. It's not that simple. It's not that just flooding parts of the brain with dopamine will cause the person to feel good. Dopamine is involved in many drugs, particularly drugs that are at high risk for addiction. Dopamine seems to be involved in reward. And because it's involved in reward, it seems to be involved in learning. So how we decide what actions to take, what actions not to take. And as we experience various events that are either rewarding or not, we learn what behaviors to exhibit. And dopamine is involved in that process. There's also serotonin. Sometimes you'll see it abbreviated as 5-HT. 5-HT is an abbreviation of its chemical name. We don't need to worry about it. But serotonin is involved in mood, which is why many antidepressant drugs, drugs that alleviate the psychological disorder of depression, uh, many antidepressants involve the serotonin system. Serotonin is also involved in things like sleep, certain physiological functions like eating, appetite control. And so it has some cognitive effects, mood, very complex phenomenon, and some simpler ones, sleep and appetite. Finally, we have norepinephrine. And norepinephrine is involved in alertness. We've already talked about these other neurotransmitters. And norepinephrine, like these others, has a particular job, and its job is a little simpler, a little better defined, and that is exciting the body. So we're going to talk about the nervous system, and what we're going to see is that there's part of the nervous system called the sympathetic nervous system that's involved in our fight or flight response, getting us alert, awake, ready for action. And so norepinephrine is the neurotransmitter involved in that process. Certainly out on your body, when norepinephrine is released, it causes heart rate to go up, causes pupils to dilate, causes your airways to open up, releases more sugar so it's available to your muscles. And so norepinephrine is involved in that feeling of alertness. It's also involved inside the brain in alertness and regulating things like wake and sleep, but also in activating attention when something is surprising. Norepinephrine kicks in and causes you to become more alert and pay attention to whatever is going on around you. So those are just some examples. There are, in fact, many neurotransmitters. These are probably the most important and the most famous, so that's why I've gone over them here. Real quick, a word on psychoactive drugs. So drugs are chemicals like neurotransmitters are. They have a chemical structure. They have an action on the nervous system. They affect the nervous system and cause some change in neural activity. And drugs can be classified into two categories. They are either agonists or antagonists. Now, you've probably come across the word antagonist before, especially when it comes to things like literature. The antagonist is usually the bad guy, the one who opposes the action of the main character. And so, in that sense, antagonist keeps that definition. Agonist may not be a term you've heard before, but it's the opposite of the antagonist. And so, what agonists do is they increase the effectiveness of a given neurotransmitter. So you can have an acetylcholine agonist, which means it helps acetylcholine do its job. And there are multiple ways to do that. An antagonist opposes the action of the neurotransmitter, makes it less effective. And there are multiple ways to do that. So, for example, many drugs mimic a neurotransmitter itself. Now, if that drug mimics the neurotransmitter and binds to a receptor and causes that receptor to do what it would normally do if it were the actual neurotransmitter, it's an agonist. An antagonist might mimic the neurotransmitter slightly, just enough to bind to the receptor, but the receptor won't actually do anything. So it blocks the receptor from doing its job. So that's an antagonist. It's decreasing the effectiveness of the neurotransmitter because it's keeping the receptor from doing its job, and it's keeping the actual neurotransmitter from binding to the receptor. But that's just one way for drugs to work. They can also alter the production of neurotransmitter itself. So here I've circled the synaptic vesicles that usually contain neurotransmitter. And if a drug increases the amount of neurotransmitter that's produced, it's an agonist. If it decreases the amount of neurotransmitter that's produced, it's an antagonist. It's keeping the neurotransmitter from doing its job. It can affect what we call reuptake. And here I've circled in this diagram a molecule on the presynaptic neuron whose job is to pull neurotransmitter back into the neuron. So neurotransmitter is released, but it doesn't stay out there forever. If it did, it would just constantly be activating that postsynaptic neuron. So one way to get rid of all that neurotransmitter is to sort of vacuum it back up into the presynaptic neuron. And that process is what we call reuptake. So if a drug increases reuptake, It's an antagonist because you're pulling neurotransmitter back into the presynaptic neuron, and it can't be out there doing its job. If you block reuptake, if you inhibit reuptake, then neurotransmitter stays out there in the cleft for longer. It can do its job for longer. And so that would be an agonist. If you've ever heard of an SSRI, a selective serotonin reuptake inhibitor, that is the kind of drug that inhibits reuptake. It keeps serotonin out in the cleft for longer. So it is a serotonin agonist, and it's a kind of antidepressant. You can affect breakdown, and there's not part of this cartoon diagram that correlates to breakdown, but there are enzymes in the synaptic cleft whose job is to destroy the neurotransmitter. Again, you can't have the neurotransmitter out there forever. We just keep signaling that postsynaptic neuron. So you can pull it back in via reuptake, or you can have an enzyme out there break it down. Now, if you inhibit the breakdown of the enzyme, that's an agonist. You're keeping the neurotransmitter out there longer because it's not being destroyed. You're therefore helping the neurotransmitter do its job. If you cause more of this enzyme to be produced, or you help that enzyme break down neurotransmitter somehow, that'd be an antagonist because you're increasing breakdown of the neurotransmitter, in which case it can't do its job. So that's just another way that drugs can have their action. Okay, that will do it for our unit on neurons. Next time, we'll move on to the organization of the nervous system at large. So we'll talk about the peripheral nervous system, which is the portion of your nervous system that's out in your body. Of course, it's all in your body, but we mean the periphery, your limbs, your torso. We'll talk about the central nervous system, which is the brain and the spinal cord. And in particular, we'll talk about the structure of the brain. What are the brain's different parts? What do they do? What are their functions? And then we'll also talk about how the brain develops. We're obviously not born with a fully developed brain. So how do we get from a small collection of cells as an embryo to a full-grown adult brain? And what role does genetics play in development and in determining the traits that our nervous system exhibits? So we'll talk about that next time, and I will see you then.